Hello, everyone, and welcome. Today, we're going to walk through how beta-phosphoglucomutase, or beta-PGM, carries out proton-driven phosphoryl transfer and why this enzyme is uniquely suited for capturing a transition state-like structure. Energy and sugar phosphates in metabolism. In biology, energy is often transferred through phosphoryl groups from one molecule to another. Enzymes that transfer or interconvert phosphate groups on sugars like beta-PGM aid in deciding where glucose goes in metabolism. As you can see in the image, the R group, or the glucose, changes which oxygen of the phosphate molecule it is bound to. Background information for beta-PGM. The enzyme we will be focusing on in particular is beta-PGM and has been recognized as a fundamental component of cells. It is essential to energy metabolism in all cells and also particularly to the synthesis of bacterial cell wall polysaccharides. There are two classes of PGMs, alpha and beta. These classes describe their specificity for the alpha and beta forms of glucose 1-phosphate. Beta-PGM is a member of the HAD family, the haloalkanoic acid dehalogenase family. Members of this family share similar residues, especially a catalytic aspartic acid that forms a transient phosphoaspartyl intermediate. They often require a cofactor, such as a magnesium ion. Initially, members of this family have been observed to adopt a related protein fold or orientation with a substrate. What makes beta-PGM different? The enzyme is a fundamental component of all cells, where to facilitate phosphoryl transfer, the substrate, glucose 6-phosphate, moves towards the active site, aspartic acid A, DA, where it is positioned in the optimal orientation. It catalyzes the conversion of beta-D glucose 1-phosphate, or G1P, to D-glucose 6-phosphate, or G6P, forming beta-D glucose 1,6-bisphosphate, or beta-G16P, as an intermediate. Specifically, researchers found that the orientation and position of the substrate flip in the active site as the reaction interconverts G6P and G1P. This geometry is also ideal for observing a transition state structure. Methods. First, the researchers built a simple backbone model of polyalanine into the electron density and then added the real side chains. Only after the protein model looked good did they add magnesium ions and phosphate where there was strong, clear density in the active site. They refined the model and checked quality using R work over R free for the Ramanakandran plot to make sure that the final structure was reliable and not overfitted. Researchers were able to experimentally trap a phosphorylated magnesium ion-bound active site that closely resembled the transition state. Selenomethionine from Lactococcus lactis PGM 1,2 was grown and purified from E. coli. The selenium was swapped in place of methionine so that it would be easier to see in the 3 wavelengths used to create the X-ray data. Methods. Selenomethionine plus MAD phase-in, the 3 X-ray wavelengths, lets the researchers solve the structure with clear active site density. The Ramanakandran plot showed that 98% of the 221 residues fell within the most favored region, which is consistent with a very stable and stereochemically sound structure. What's especially interesting is that the researchers captured beta-PGM in a state where aspartic acid 8 remains phosphorylated, but the bound G6P appears to have dissociated and rebound in a transition state-like orientation. This gives a rare glimpse at how the active site organizes itself during catalysis.
Right. So the structural results from our X-ray crystallographers and the researchers' experiments showed that there are several core domains, or several domains. There is an alpha-beta core domain, and for structures like beta-phosphoglucomutase, the expected HAD fold has many of these common alpha beta core domains, which serve as the main catalytic center. In addition, there is also a cap domain that sits over the active site and helps the substrate binding and closure. Aspartic acid 8, in addition, also lies in the center of our active site pocket between our core, as shown in blue, and our cap, as shown in orange. Along with that are our magnesium ion, as shown in dark blue, and the substrate, as shown in yellow. These residues around the active site match conserved HAD family motifs, which support a mechanism where aspartic acid forms a phosphorylated intermediate during catalysis. In addition to the domains observed by our researchers, there are 6 stranded parallel beta sheets and 6 alpha helices in the core domain. Alternatively, there are 4 helix bundles between beta 1 and alpha 5, comprising the cap domain. Another key result from the paper is the comparison between the apostructure of the protein, which is without the ligand or substrate, and the ligand-bound or intermediate-like structures. So in the apo form, with no ligand bound, the active site is more open and the cap and loop regions are pulled away from the pocket. In the ligand-bound or transition-like structures, these elements move inwards and close over the active site. In this closed conformation, the aspartic acid 8 and magnesium ion and the sugar substrate are all lined up correctly to allow for effective catalysis. These conformational changes support a mechanism where substrate binding causes active site closure, stabilizes the transition state, and makes phosphoryl transfer more efficient. Oh no, something just happened. Okay, we're good. Sorry. In a model, in a modeled beta phosphoglucomutase active site, as shown here, the sugar is positioned so that the C6 hydroxyl group, shown here, points towards our aspartic acid 8 residue here, where the same carbon is also close enough to our aspartic acid 10, allowing for proton transfer to occur and then forcing phosphoryl transfer to move over to our aspartic acid 8. Additionally, a comparison of the ground state, beta PGM G6P complex with the trapped transition state showed that the active site recognized during showed how the active site is recognized during catalysis. So in a first step, the aspartic acid 8 acts as a nucleophile attacking the phosphoryl group of the glucose 6-phosphate. Because it is in line with the aspartic acid 10 group, this is able to act as a proton donor to the glucose 6-phosphate as it leaves into solution. So in this second step that is not shown here, this phosphate group is transferred back from aspartic acid 8 to the sugar at C1, generating the glucose 1-phosphate. Aspartic acid 10 is positioned to function as a general acid base and may serve as a second nucleophile in a 2-nucleophile mechanism, which was observed, but it was not recognized as the most efficient mechanism for the proton-driven phosphoryl transfer in beta-phosphoglucomutase. The observation of this trapped intermediate or phosphoaspartic 8, along with the magnesium ion, non-covalently bound um, in the crystal structure, provides strong structural support of this phosphoryl transfer pathway. So additionally, the researchers observed that lysine 145, serine 114, and the magnesium ion helped stabilize the negative charges within the active site and orient the reacting groups. Polar residues around um, the sugar form two sides. One side includes serine 114, lysine 145, aspartic acid 8, and glutamic acid 169, as shown in blue. The other side, um, which aids in proton transfer, shows thiamine 16, aspartic acid 10, lysine 45, and aspartic acid 170, which is shown in pink. Between the two sides, with the pink supporting proton transfer and the blue supporting phosphoryl group transfer, we are able to implement the ideas of flexibility that is required in the active site for this proton-driven phosphoryl transfer. And we are predicting that serine 116 and lysine 117 helps tune the balance between flexibility and stability near this cap core interface, as shown in yellow. So some key takeaways from what our researchers have been able to accomplish. Beta-phosphoglucomutase is a HAD family enzyme that interconverts glucose 6 phosphate and glucose 1 phosphate via a phosphorylated intermediate on aspartic acid 8. The structure of phosphorylated beta phosphoglucomutase captures a transition-like active site with aspartic acid 8 and phosphoryl complex, along with the magnesium ion. The alpha beta core domain and cap domain and the conformational changes between open and closed states work together to align aspartic acid 8, the metal ion, and glucose 6 phosphate for efficient proton-driven phosphoryl transfer. These results combine structural observations made from newly formed crystal structures from the researchers and a mechanistic reasoning to give a detailed picture of how beta-phosphoglucomutase controls phosphate movement on glucose in cells. Here are some of our acknowledgements and our primary source. We would like to thank PyMol and UnitProt for the data and for being able to use it for data collection and visualization in this webinar. Thank you.